This is a review of selected questions that the class did not perform as well on in Module 2 quiz Part B. I'm going to skip down and find some that were not performed well and only present those to save time. Okay, this is one of them, question 2, and at least a question in the, it won't be question 2 in your quiz, but several examples of structure when encircled and select a correct identification. Well, normally contain a blood vessel. That was the correct answer, but only 36% of the class got it correct. Some people thought it was lacunae and hyaline cartilage. Well, first of all, you need to recognize it was bone. And so here's the low magnification over here, and you could have zoomed this out like this and even locate maybe some skeletal muscle on the side here. So zooming out, you may recognize adipose tissue, and this is bone marrow with some hematopoietic cells in it. Over on the side, this is skeletal muscle, and in between you have this intensely eosinophilic tissue that when you enlarge it, you see these patterns. And these patterns, you have a substance in the center, and then you have these concentric layers around it with nuclei. Well, this is compact bone, and the question actually referred to these structures. Here's one that's empty. Here's one that has some blood in it. Here's one that's almost closed, and this one's almost closed. So you were asked, something about that, and it contains a blood vessel. That's a Versian canal in compact bone in the center of an osteone. For this question, about 65% of the class got it correct. Select a true statement about the intense blue staining ring-shaped region in this virtual slide specimen. The correct answer has the most dense concentration of proteoglycan molecules in the tissue sample shown. Well, it was cartilage. It was hyaline cartilage. And you can see the matrix here, the interstitial matrix, that's a little pink and less blue. And then right around the lacuna where the chondrocytes are enclosed is this intense basophilia stain. And the proteoglycans are basophilic. So that is the reasoning behind that. This is perichondrium out here. This is chondrogenic, where you've got the stem cells and new chondroblasts being formed, which are divided and eventually form chondrocytes. And this is the fibrous portion of the perichondrium. So this question asks, what describes the processes occurring between the two dotted lines? And the correct answer was lengthening of the diaphysis of a long bone. Well, not very many of the class got it correct, but it's pretty clear. This is the epiphysis, where you have hyaline cartilage, and then you've got the cartilage dividing in here in this area and then enlarging. And if we enlarge this, you will see intense mitosis taking place here, and then the cartilage cells getting larger here, and then you get some calcification of cartilage here, and then the deposition of bone down here. And this is bone forming here. So what was happening there was the lengthening of the diaphysis, and what you were looking at between those two lines is the epiphyseal plate. Epiphysis, diaphysis, epiphyseal plate, adding to the growth of a long bone in length. And this question was asked about a tissue region between a black dotted line and the space indicated by two arrows. And the region was the zone of tangential gliding articular cartilage. Now the terms itself are fairly descriptive. Tangential means like a tangent of a circle, and gliding would be close to the articulate surface of cartilage. And that's what it is right here. This is the epiphysis of one bone. This is the epiphysis of another bone. And in between, you have the articular surface. And you were instructed in the lecture that this would be the tangential gliding area of the cartilage or the articular cartilage. Okay, here's a question where a specimen of a bony spicule was presented, and the correct answer was a lamella and bony spicule. You were asked to choose the answer that best describes the alternating light and darker cytophytic bands in the structure between the two dashed lines. So you had to recognize it was bony spicule and not cartilage and not compact bone, because there were no Haversian systems there. And here's the specimen. You see it's a spicule. If you look at it low magnification, there it is. This is compact bone up here with Haversian systems, you know, with Haversian canals, etc. But this was a spicule. And these alternating light and dark bands, those are lamella. You do have lacuna and osteocytes here, but they're not arranged around a Haversian canal in a spicule. Okay? Okay, this question you were asked with the normal fraction of this cell, the percentage of the cell in a differential leukocyte blood count. And you were given, this is a very low percentage, 0 to 0.75%. And 67% of the class chose the correct answer, and that was a basophil. And basophils are very, very scarce in peripheral blood. But you can see the nucleus, and it appears to be bilobed, but the most dramatic thing about it are these basophilic granules that you could have enlarged more if you wanted to with empty magnification in case you couldn't see those granules well. See there, you see them quite well, and the background or the shadow of the nucleus. So I'll return that to the proper magnification. A basophil, very low percentage. It just reminds me to remind you to review the percentage of white cells in a differential count. Okay, this question said within the two blue lines is a component of one of the tissues you have studied. We wanted you to name the component defined by the two blue lines. Well, the correct answer was periosteum. You could have said fibrous periosteum, or you could have said osteogenic periosteum. But you had to answer periosteum. And only 45% of the class got it correct. This is bone. You see this is developing bone here. The Haversian canals are quite large, but you can see the beginning of the deposit of the bone around a concentric layers around the Haversian canal. And so you had to recognize this was bone, and this was connective tissue. And you didn't have to, but out here is smooth muscle. And then in here, this is actually bone marrow with some hematopoietic tissue and some adipocytes. But you were asked the tissue on the edge of the bone. And that connective tissue is periosteum, and those cells right here, that would be the osteogenic portion of the periosteum, where you have the stem cells that produce osteoblasts, and these are collagen and fibroblasts in the fibrous periosteum. Okay. This question asks you to identify the cells indicated by the blue arrows, and they were megakaryocytes. You could have said megakaryocyte singular or megakaryocyte plural. Only 56% of the class got that correct. And here, you know, you could take this down to low magnification and see we used the same specimen as the previous question, and this is bone marrow. And so if you go to the magnification we set it at, you can see that these cells have an eosinophilic cytoplasm, and they have a huge, large nucleus. And it's not multinucleated, and the fact that they are sitting right in the middle of the bone marrow. If it was a osteoclast, it would be on the edge of the marrow next to the bone in a Howship's lacuna, because that's where it does its function. And a osteoclast has multiple nuclei, whereas a megakaryocyte has one huge nucleus, and these are the cells that produce the little fragments that end up in the bloodstream called platelets. So that concludes the review of Module 2 Quiz Part B.